and organize. So thank you for the introduction and organize this wonderful journal club. So I'm Shoutang Wang, a postdoc working at Dr. Corona's lab at Washington University of School of Medicine in St. Louis. So today I'd like to introduce one of my projects related to the function of microglia in Alzheimer's disease. So my presentation title is Anti-Human TREM2, Introduced Microglia Proliferation and Reduces Pathology in an Alzheimer's disease model. So Alzheimer's disease, short for AD, is the most common form of dementia that mainly affects aged people. So AD is characterized by memory and executive functional deficit, followed by global and progressive cognitive declines. AD pathology, brain AD pathology consistent of extracellular aggregation of amyloid beta, short for A-beta oligomers, large insoluble plaques, and intracellular tau hyperphosphorylation, as well as neural cell death. AD lesions also trigger a secondary expansion of active microglia, which cluster around A-beta, limiting their spreading. So study of genetic risk for sporadic AD have suggested that microglia not only responds to disease, but also moderate disease course. Most notably, TREM2, among other genes such as ApoE and BM1, was addressed as a higher risk genes for acceleration of AD pathology processes. So TREM2 is an immunoreceptor with extracellular Ig-like domain, a stalk region, a transmembrane region, and a short cytoplasmic trails. TREM2 was found like only expressed by microglia in the central neural system. And it has been shown that in vitro, TREM2 can bind HDL, LDL, ApoE, LPS, and other polymers, such as nucleic acids and A-beta. So by using 5FD mice, a mouse model of A-beta accumulation, our lab previously showed that TREM2 deficiency prevented microglia clustering around the plaque. TREM2 deficiency also alters plaque density. As I show here, plaque from 5FD mice were compact with condensed core with few surrounding diffuse structures, while plaque from TREM2 knockout 5FD mice were more loosely packed with smaller cores and many fiber-like structures extending outwards, which result in increased axon swelling and neurite dystrophy tested by N-terminal of APP staining. Moreover, a recent single-cell RNA-seq analysis of microglia in 5FD mice showed that the microglia transition from a homeostatic pattern to a degeneration or disease-associated pattern, which we call this DAM. Microglia transition from the homeostatic to stage 1 DAM in a TREM2 independent fashion. And then microglia switched from stage 1 DAM to stage 2 DAM through TREM2 mediate signals, suggesting that generation of terminal stage of DAM in 550 mice requires TREM2. So based on what I showed, based on above description, we were wondering why the treatment of an agonist anti-TREM2 antibody is beneficial to AD pathology. So in this project, I used human TREM2 common variant, short for CV, and mutated R47H transgenic mice crossed with 580 mice. So for this mouse model, we previously showed that the R47H mutation causes a marked decrease in microglia activation markers in 550 mice and impairs microglia clustering around the plaque. So the first part of the project is to validate the anti-human TREM2 antibody AL002C to test the specificity of the AL002C. We stand bone marrow derived macrophage prepared from CV and R47H mice. We found that the AL002C bind the human TREM2, and the binding is not impacted by the R47H mutation. So to test whether AL002C activates human TREM2, we next examined the calcium-driven cell reporter 2B4 stably transfect with the human TREM2 or R47H And in these reporter cells, engagement of TREM2 promote generation of GFP, which can detect by flow cytometry. So we found that AL002C induce GFP in the both CV and R47H genotypes, while IgG control did not, suggesting that AL002C acts as an TREM2 agonist. So we next examined whether AL002C impact in vitro survival of macrophage after CSF1 starvation in ATP level based survival viability assay. We found that treatment of AL002C significantly increased macrophage survival, suggesting that AL002C activates both human TREM2 variant. So to characterize the impact of AL002C on microglia activation, we performed the single-cell RSeq. So 5 months old of CV and R47H550 mice were injected by the AL002C or control IgG. After 48 hours, the CD45 positive cells were isolated from the whole brain and submitted for single-cell sequencing. So taking advantage of single-cell revolution of our data, we reclustered microglia into 12 clusters, which include homostatic microglia, DAM, interferon response microglia, proliferating microglia in S and M phase, as well as microglia with intermediate features between homostatic microglia and DAM, which we defined as transitioning microglia. Further examination revealed that cluster 7 and cluster 9 were significantly enriched in AL002C treated mice. GO term enrichment of biological processes showed that The cluster 7 is enriched in genes encoding ribosome proteins, indicating metabolic activation. And cluster 9 is enriched in cell cycle genes, indicating a higher proliferation rate. So we conclude that acute engagement of human TREM2 on microglia by AL002C promotes a signature of metabolic activation and proliferation. So the antibody was nice to test in the 550 mice for a chronic treatment. The CV and alpha-7H 550 mice were treated with IP injection of AL002C or control IgG with 30 milligram per kilogram. To mimic therapeutic treatment, we start the injection at 5 months old when the mice have already developed the A-beta aggregation until 8 months old when the A-beta is still growing. We also perform the behavior test during the treatment. The mice were finally sacrificed within 48 hours after the last injection. So we first examined the impact of AL002C in in A-beta plaque compaction. As I mentioned above, TREM2 deficiency influence plaque density. Technically speaking, the filamentous plaque can be detected by the brain staining with 6E10, which identifies the A-beta aggregates, whether they have beta sheet or no beta sheet conformation, and considers it's more neurotoxic. Inert plaque comprise A-beta aggregate that form a compact core with no A-beta radiating into the surrounding parenchymas, which can be labeled by metoxyxo4. The compact plaques displayed 6E10 positive A-beta and compact metoxyxo4 cores. So by using this inflorescence approach, we found that compared to control group, the administration of AL002C induced overall reduction of filamental plaques across the CV and alpha-7H genotypes, and increase the percentage of inert plaque, whereas the number of compact plaque are similar between two groups. So given the differential neurotoxicity of filamental inert and compact plaque, we next examined whether changes in plaque perform impact the neural damage. So we stain the brain section by N-terminal of APP, which accumulates in the dystrophic neurites, and measured the APP positive neural damage, neural precise around the plaque. So we found that compared to the control group, the AL002C treatment significantly reduced the volume of dystrophic neuron per plaque. So we next examined whether reduction of A-beta pathology impact or associate with behavior changes. So one 5FAD and alpha-7H mice were tested in the elevated classmates the total number of entrants and the distance traveled in the open arm were significantly decreased in both genotypes. We next directly examined the impact of AL002C on microglia. So we measured the cell body size and the total length of precise which the microglia monitor their surroundings. We found that in AL002C, interestingly, the two parameters were significantly increased in the cortex and the hippocampus of AL002C treated mice compared to the control group, suggesting increased surveillance activity. Moreover, we stained microglia for CD68, a marker of activation and phagocytosis, as well as A-beta 42. 3D reconstructors with A-beta 42 and CD68 immunofluorescence revealed that the AL002C treatment significantly increased the ratio of CD68 A-beta 42 double positive microglia phagosome over total A-beta 42, suggesting that anti-TREM2 promote microglia activation and phagocytosis of A-beta. So we finally stained the brain section for IBA1 and marker of inflammation, SPP1. And we found that the AL002C significantly decreased the expression of SPP1 in microglia in the both genotypes. So to summarize what I showed here, a single injection of the human TREM2 agonist antibody AL002C in the human TREM2 transgenetic 550 mouse model acutely expanded microglia subsets that are metabolically active and proliferate. Prolonged systemic administration of AL002C reduced fundamental A-beta plaques and neuritis damage, settled but significantly changes in risk-taking and anxiety-like trials preload the reduced pathology. Inflorescence analysis of microglia showed signs of increased surveillance activity after chronic AL002 treatment. So we finally conclude antibody-mediated engagement of human TREM2 enhances neuroprotective microglia, restricting neurotoxic A-beta plaques and neurodamaged. So we'd like to thank our collaborators from Elector, which contribute a lot to the project. And also I'd like to thank Professor Claire Yuda in the university, in the Washington University, who helped me perform the behavior test. So thank you for your time. Okay, that's it.
Thank you. Thank you, Shoutang. That's very important work. So I guess we have a few questions in the chat box already. Could you click on the chat and repeat the question, please?
Okay, give me a second.
Yeah.
Okay.
It's probably easier if you stop sharing.
Okay.
Or I can read them for you.
Yeah, I think so.
Okay, yeah, okay. So the first question is, nice talk, 2 questions. When you isolate the CD45 population, did you specifically specifically choose CD45 low or CD45 high or all CD45 positive cells?
Well, yeah, I sought all the CD45 positive cells because at the beginning we were interested to analyze all the immunocells, not only microglia, because this is a system, the IP injection. So the antibody might impact the other cells, other immunocells. So we include everything.
Okay, the second question is, have you checked your antibody effect on the other CD45 population in addition to microglia?
Yes, we checked, but we didn't see any change.
Okay, so I have a follow-up question. Have you also checked astrocyte activation or other brain neuroinflammation markers such as pro-inflammatory cytokines in the antibody-treated mice?
No, which we should, but no. Yeah,
Okay.
we will do it. Yeah.
Yeah. Okay. So there's another question. Have you tried any specific microglia-specific antibodies? Sorry, I'm not sure what exactly this means. If Person Max, you want to elaborate a little bit more?
Yeah, please.
You're welcome to turn on your video and speak. Meanwhile, maybe I'll move to the next question from Jennifer. Very nice, thank you. Her question is, were the microglia containing A beta after treatment localized to filamentous plaques?
Were the
Were
containing A beta after treatment localized? I think the microglia contact the A beta not only for the filamentous plaques. I think it's when I tested the CD68 staining, I found that all the type of plaques. Yeah. I checked all the. I didn't pick up the specific filamentous plaques, or I just pick all the plaques, and I think all the for all the plaques is the same.
Okay, there's one more question from Shoutang. Were the disease-associated microglia specific microglia population dramatically changed in your assay?
Well,
CD45 low, CD11C positive.
Well, for the DAM, we didn't see, or firstly, because this is acute, the single cell RC was performed after acute injection, only 48 hours. So it might be different for different treatment. So for this analyze, we didn't see the significant changes for the percentage of the DAM. But we do see some DAM markers increased after the antibody treatment.
Okay. So the next question from James, very interesting, thank you. His question is, do you think the increase in metabolic activity and proliferation in wild-type mice would have any observable effect on microglial activity in non-disease state?
Yes, because I didn't show here, we also test, we also inject the antibody to wild-type mice, and after 48 hours, we do see, and we did the we did the Ki67, and we do see that the antibody treatment increase the percentage of Ki67 positive microglia, which means that the microglia generally increase the proliferation, not only in the disease model, but also in the wild type.
I see. Okay. Thank you. So we're waiting for more questions from the audience. I have another one. Also gave AL002C to 5-month-old 5 safety mice, and you treat them for 3-month long.
Yes.
So if the antibody was safe and effective for human patients, so in your opinion, what would be the best time to treat AD patients?
Well, to be honest, this is just the small piece of the, I think this is just a small piece of the project. There is, to be honest, personally, I think there would be different or plenty of possibilities for this if you want to translate the experiments in the mouse model to the chronic treatment. I think it will have a really long way to go. So in our experiments, we showed that the antibody treatment is benefit when the start stage of the disease, saying that because in the 5 months old, the disease, the mouse model has already have the A beta aggregation. So this might work, but we don't know whether after the late disease process, what's going on for the treatment, saying that 1 years old 550 mice, we don't know. So it will be interesting to try the different possibilities.
Yes, for sure. Okay, so do we have more questions from the audience? Oh, yes, there's one more. Could you explain more why you use the acute treatment for single-cell RNA-seq instead of the long-term treatment?
This is a good question because we are going to do the single-cell RNA-seq after the chronic treatment. At the beginning, we want to know the reason why we do the acute treatment is because at the beginning we want to know know more the mechanism of this engagement of TREM2 in vivo. So yeah, we are going to perform the long-term single-cell RNA-seq.
Okay, so it's good notes on in the pipeline.
Yeah.
Okay, so any more questions? If not, we'll